I am pleased to be hosting Dr. Giulio Di Minin to discuss his recent PLOS ONE Lab Protocol paper entitled Applying Spinal Cord Organoids as a Quantitative Approach to Study the Mammalian Hedgehog Pathway and the Associated Detailed Protocol in Protocols.io. Dr. Di Minin is a dedicated scientist with a profound understanding of signaling networks in stem cell biology. He pursued his PhD at the University of Trieste in Italy, supported by a fellowship from the Italian Association for Cancer Research. To further expand his expertise, Giulio joined the laboratory of Professor Alan Frankel at the University of California in San Francisco. Subsequently, he received a Marie Sklodowska-Curie Actions ETH Fellowship to conduct his postdoctoral research in the laboratory of Professor Anton Woods at ETH Zurich. During this time, he applied mouse haploid embryonic stem cells in forward genetic screens, leveraging their unique haploid genome to achieve the highest screening resolution in mammalian cells. He applied this cellular system to investigate the regulation of the Hedgehog pathway during stem cell differentiation and how its deregulation can lead to the development of aggressive tumors. Currently, as a senior scientist at ETH Zurich, Giulio is embarking on establishing his own independent research program. His primary objective is to exploit the plasticity of haploid stem cells to generate neural stem cells models and to use advanced forward genetic screening strategies to investigate the events promoting brain tumor formation. Giulio, thank you so much for being here with us.
Thank you, Gabriel, for this nice invitation and also for the introduction.
So to kick off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
And so, as from the title, the protocol focuses on the application of spinal cord organoids as a cellular system to investigate the hedgehog signaling pathway. So, for understanding its relevance, is first of all important that I briefly introduce you to this peculiar signaling cascade. So the hedgehog pathway plays a crucial role during embryonic development and also in tissue regeneration in adults by controlling the homeostasis of stem cells. Unfortunately, another reason why the hedgehog pathway is important in adults is that it's frequently found hyperactivated in a variety of different tumors. So a better understanding of the hedgehog cascade is not just relevant from a developmental perspective, but is important for better understand what are the processes that are leading to the regulation of this signaling and to the process of transformation. One of the processes in which in development the hedgehog pathway plays a crucial role is during neural tube specification. So when we are talking about the neural tube, we are talking about a tissue of the developing embryo that leads to the formation of the brain and the spinal cord. So here, an hedgehog gradient is crucial to define the specification of different neuronal subtypes, 5, 6 different ones. So as you can understand, when we are talking about the hedgehog pathway, we are not discussing off and on-off system, sorry. But we are discussing of a signaling that according to its intensity leads to different cellular and transcriptional responses. We can easily monitor this different type of response, analyzing during neural tube specification using like a readout markers that are specifically expressed by these neuronal progenitors. For example, we have the more ventral neural progenitors that are leading to the formation to motor neurons that are expressing NKX 2.2, and that depends a lot on hedgehog hyperactivity for their specification. Or we have more interneurons that are expressing the PAC6 marker, and that instead depends less on the hedgehog signaling for their specification. So the spinal cord organoids are able to mimic in vitro these complex specification events. So if I can go a little more into the methodology, maybe skipping some of your questions. The starting point are embryonic stem cells, cell lines that can be easily expanded, culture in any kind of cellular system, and that have the property that can be easily edited, meaning that we can introduce overexpress a factor of our interest, or we can deplete another one using a strategy, for example, CRISPR-Cas9. So we are taking these embryonic stem cells and we are forcing them to aggregate using some specific type of dishes that are leading to the formation of some kind of spheric structures. These spheric structures are then treated with retinoic acid, a compound that is able to promote the spinal cord identity of our embryo bodies. At this point, we have our spinal cord organoids, and we can stimulate with the different hedgehog levels to promote the specification of the same neuronal progenitor that we are finding during neural tube specification. As you can clearly understand, we have now in our hands a tool that allows us in vitro to modify our system with the perturbation that could be kind of chemicals or also genetic approaches, and analyze how these perturbations are affecting the hedgehog signaling cascade for mechanicistic purposes.
And so what was the motivation? And you already explained it briefly during the previous answer to develop a protocol. In other words, can you elaborate on what was the state of the art when you decided to optimize this protocol, and how people before your protocol tackled these questions?
Okay. So I'm only starting from long ago. So the hedgehog pathway has been discovered more or less 50 years ago. And beside a lot of knowledge that we succeeded to acquire on this, the regulation of the signaling cascade, we are still missing the understanding of a critical step of how the signaling is transduced. Just one example for helping you understand what we are lacking. We know that the hedgehog receptor, patch, is a transporter, but still we don't know which is the molecule that is controlling and that prevents hedgehog signaling activation. So we don't know which is the key signaling transduction of the signaling cascade, and we neither know where this regulation occurs. There are some studies indicating the primary cilia, other studies indicating endostatic vesicles. So this is just an example, but there would be plenty of other ones. Try to describe all the holes that we have in our knowledge of the hedgehog cascade. And if you ask to me, one of the reasons why we are limited in studying this signaling cascade is that we are lacking proper cellular system to mechanicistically dissect it in mammalian systems. We can perform our experiment, for example, in vivo, so using embryos and analyzing neural tube specification process. But as you can clearly understand, using the in vivo system for addressing mechanicistically studies is unfeasible. Or we can use fibroblast cells. Those are cells that can be easily expanded, edited, and that are responsive to Hedgehog signaling. However, when we are using this type of cells, you need to consider that we are talking about terminally differentiated cell lines that to become responsive to Hedgehog signaling required to be deeply starved and also cell growth arrested. How can a cell system like that mimic the complexity of regulation of a cascade that instead promote cell proliferation and control cell differentiation event? I mean, it's quite doubtable, I would say. So spinal cord organoids represent a kind of a middle ground situation between this opposite scenario, offering the in vitro benefit for mechanicistic studies, but allowing us to study the regulation of the Hedgehog pathway at extremely specific level. And we are missing in the field the application of this peculiar system for investigating the Hedgehog signaling cascade.
And now, like going a little bit deeper into the protocol itself, what do you think are the most critical steps? Those in which the user of the protocol really have to pay attention.
So definitely for the protocol, you need some experience in stem cell capture. Okay, but beside that, I would say that the protocol is quite pedestrian, so there are not something extremely complicated. There are two steps that I would mark like, and that actually we mark during the lab protocol like crucial, that are these spheric structure formation, so they need to be of a specific size for further developing to spinal cord organoids. And then is the treatment with retinoic acid. So the retinoic acid is quite crucial, as I briefly mentioned, for promoting the spinal cord identity. However, this compound is quite unstable and also is quite powerful. So changing of little its concentration will lead to different effects. So for this reason, in our protocol, we are suggesting when you're trying to develop this procedure that you need to try a different range of retinoic acid, at least at the beginning, to define, according also to the batch, how you're storing the compound, which one is the optimal one working for your specific environment.
As you were developing and optimizing the protocol, was there anything that you tried that didn't work and didn't make it to the final version of the protocol?
And do you have all the night to discussing about? No, I'm joking. For sure. I mean, when you are developing a protocol, you are trying different approaches, and I mean, some are working and are going at the end to the final protocol, and then there are others that maybe were extremely brilliant, but they are not making sense from a biological perspective. One of them was exactly connected to the retinoic acid part. So we tried to use a different strategy considering this instability of the product. And we tried to use, so the retinoic acid is important for neuralizing and caudalizing our spinal cord organoids. And we tried to split these two different effects using two compounds, so and media that should support from stem cells their neuralization, and then we try to use a Wnt inhibitor, chiron, for promoting the caudalization of our neurostem cells. However, we tried quite hard this system, but never led to the same result that we were obtaining with the retinoic acid. And so at the end, we prefer to remain strict on the retinoic acid application.
So we talk about the motivations to develop the protocol and where the field was. And we have talked a little bit about, the steps of the protocol, what is important to really focus attention. I would like now us to change gears a little bit and discuss about your motivations to have this protocol peer-reviewed as a standalone publication. And if I recall correctly, you used a version of this protocol back in 2022 in a paper that you published in PLOS Biology. And then you took the protocol itself and decided to peer review the protocol and get it published as a lab protocol paper in PLOS ONE independently of that PLOS Biology paper. Can you elaborate on what was your motivation?
Yeah, sure. So this methodology was essentially developed to validate candidate genes identified in an haploid screening. So my expertise, like you mentioned during the introduction. And the paper that you described, that you cited, the PLOS biology paper, is essentially centered on the description of this screening procedure and the validation and characterization of one of the candidates that we identified, TIMET2, that we found to be extremely crucial for the regulation of smoothen, another key player of the cascade, regulating its secretion from the ER Golgi compartment. So considering that the TIMET2 knockout embryos that we obtain were not able to develop until the proper specification of a neural tube, we used spinal cord organoids to a little more in a more physiological context, validate the role of TIMET2 as a repressor of the Hedgehog cascade. Naturally, as you can imagine, in a paper like that, we didn't have all the space and the time for going into the detail on the protocol characterization that we developed during the process. However, going to different Hedgehog meetings, what we noticed was quite some kind of interest for the result that we were providing, and particularly a curiosity for the methodology, for how we were applying spinal cord organoids for investigated Hedgehog signaling cascade. So for this reason, together with the PhD student working with me, Marcus Holzen, that actually is the first name in the PLOS ONE paper, we decided to better describe the methodology in an independent lab protocol, such that we hope to support the Hedgehog field.
And once the protocol was accepted for publication, why did you decide to make the peer review history public?
I strongly believe that the revisions process is not just a stamp that you're adding to your paper and that is necessary for getting accepted. So the revision process is a crucial part of the developing of your paper and the developing of your history, of your story. Also because it's the first time that you are getting exposed to comment, in many cases, of people that are expert in the field, and with their comment can help you improve the value and also the significance of the story that you are telling out. So generally, when I'm reading a paper found somewhere else, I also like to go through all the revision details because I'm curious, but also because helps me to understand what are the strengths and also the weakness of the paper that I'm reading. And the second, but not for importance, going through these details helps me quite a lot in crafting better the responses for my own paper. So, I mean, I'm using the paper of others, so I should also provide what I went through.
And can you summarize a little bit this conversation between you and the peer reviewers and tell us how their feedback helped you shape the final version of the protocol?
So at the beginning, the reviewers were a little critical, so it would be interesting for you to go in this case to read all the comments. So they were a little critical regarding the novelty of the approach. So they were pointing out that spinal cord organoids have been applied for a long time for studying neurostem cell specification in vitro. And they were perfectly correct in that. I mean, there are top labs that are using spinal cord organoids and developing spinal cord organoids to categorize neurostem cell specification for weeks and even months. But with our protocol, we didn't want to interact, to provide a tool for the spinal cord organoids community. Our intention was to provide a tool for the Hedgehog community. So what we are describing here is the application of spinal cord organoids as a readout for analyzing better the Hedgehog signaling cascade. And for this purpose, our attention was to some details in the procedure such that were practicable for this specific purpose. So we tried to develop an approach that is fast, so it's not requiring too many days and not months, because you don't want to wait months before getting a result. So we are getting the final analysis in 5 days, and then we work quite strongly to make our spinal cord organ information quite reproducible, such that we don't have different signaling effects between replicates. And importantly, we provide different ways, different approach to quantify the Hedgehog signaling pathway. So there was kind of a misunderstanding, and the comment of the reviewer were quite helpful to highlight this misunderstanding that we are correcting in the final version of our manuscript. And after that, they also agree that the protocol is quite an interesting approach for the Hedgehog signaling cascade. And in fact, they also suggest a couple of experiments for further improving the methodology. And we went through quite interestingly through their comments. And if I'm correct, according to their suggestion, we added 3 additional panels of new experiment in the final version of our manuscript. So was quite a satisfactory revision, I would say.
And I just have one more question. So I would like to know, what has been the significance of publishing this lab protocol paper for your career?
So as you mentioned in the introduction, I'm from a career perspective in a phase in which I'm trying to develop my independent group. And naturally, one crucial aspect of being a principal investigator is also to getting your story published. So this lab protocol, sentimentally, has been the first paper in which I behave like last senior co-author, how you wanted to call it. And I have to tell you that there was quite a formative experience having to deal directly with editors, reviewers, and also publishing methods. Another thing kind of connected still of the fact that I'm quite young in this job it helped me publishing this paper to developing also my scientific network. So after the publication of this paper, I've been contacted by other people in the field, and we started to discuss about the protocol, some kind of suggestion. Then we further develop ideas. So I believe that those kind of interaction will be quite helpful for my following career. So, I mean, guess that those are the main two reasons why I believe that it will remain always kind of in my heart, this paper. Yeah.
Giulio, thank you so much for sharing. As I mentioned before, I don't have any other questions. I don't know if there's anything that you would like to add that haven't been discussed so far.
No, I guess that we succeeded to went quite nicely through all the details. I hope that someone will find useful this kind of information and will strongly apply this system for investigating better the hedgehogs in Gasque.